Hello.
Morning. Work. I need to close this and restart it. Good morning, everybody. Trying to get things all set over here. Internet is very slowly at school this morning. Device, but we'll get there. I hope. I love it when things don't work. But that's okay. People are still popping in. Gotta wait till everybody gets here anyway, right? Alrighty, let's try this on for size. Take a second for it to load, but it should be here in just a moment. And then I back here. Goodness gracious. All right, so while we're waiting for Pear Deck to do its thing, a couple of quick announcements. I hope you saw the text this morning about assessment number 5, still a take-home. Still going to use that stance for as long as we need to, probably for the entire year, probably for the rest of my career. It's not due until Monday, so please let me know if that's not going to work in terms of your schedule. It's pretty short, you know, they're not very long in general. I think I've got 7 multiple choice questions and 3 free response questions. And of course, the page is not responsive. Here it comes. So I think there's one question that you might not be able to answer until the end of this week. It's on lipid or fat biochemistry. So if you want to absolutely hold off until the weekend and then do it, that is a strategy. But if you wanted to get a head start, just leave that tab open on your device. You could start as early as today. So that's item number 1. Item number 2, before we get started, the next time we meet, if you're coming to school, I did want to try, fingers crossed, a short in-person lab with people. It's nothing revolutionary. Really, I want to get my feet wet, but also your feet wet, working in a pandemic setting, in a socially distanced setting within the classroom. So I have that on the agenda for next time. People at home, you're still going to be able to do the experiment. Again, it's nothing fancy. I've got a virtual replacement for you that you can take a look at. So just want to give you a heads up on those 2 items as they come up this week and next week. I think Pear Deck is doing its thing now. At least I hope that it is. What questions do you have for me or for the good of the group before we get started? As always, if things pop up, don't hesitate to ask. Alrighty. So I do have a very brief check-in. I still find it hard to believe that we haven't been together for a week. So I wanted to do a really brief check-in with you this morning. Then I've got some review for us, thinking back to our last time together. We began to look at the actual reactions, the biological reactions that are responsible for anabolic processes, putting molecules together. You might remember those are dehydration or condensation reactions. But we also did some work with the biological functional groups, because those are the actual parts of biological molecules that are going to react with each other as part of anabolic processes. So I've got some brief review questions for us to take a look at. And then I have the next step. If we can put molecules together biologically, anabolically, we should be able to take molecules apart catabolically. So there's a type of reaction that we want to look at called hydrolysis. So I've got, again, some practice questions with that. And my hope is that for today, you had a chance to watch the Ed Puzzle video on carbohydrate biochemistry. I've got a couple of things, depending on how much time we have left over, to look at why this stuff matters. Again, not because you need to memorize any of those molecules, right? Or the structure of any of those molecules, or the molecular formulas of any of those molecules. That is preposterous. But to begin to look at least a little bit at why the differences between these molecules in this setting, carbohydrates, actually matters to us biologically. So that is our plan for the day. And we'll take a break in there somewhere along the middle, Okay. Alrighty. Well, again, in the spirit of making things as normal as we possibly can, at least a normal routine, it's been a week, right? Over that week, we had a break. I hope that you took a break. I know that I tried to. If you were to think back over these last 6-7 days, over the holiday, if your family celebrated the holiday, is there a word that you could use to best represent your experience? Can you write that word? Can you draw that word just as we get started today? It doesn't have to be one word. It can be a phrase if that works better for you. I think most people have had a chance to respond. People are getting knocked off too. All right. Let's take a look at the word or words that best represent your experience here. Lazy, tired, average, relaxing, chill, refreshing, needed, for sure, boring, which isn't always a bad thing, right? Relaxing, busy, calm. Reminds me to put my feet back on the floor to ground myself. Quick, it was fast, wasn't it? Relaxed, making up missed time, relaxed, memorable, that's awesome. Lonely. It's not the first time I've seen that. I too was lonely. Okay. On edge, yeah. Short, productive. All right. Well, I'd like to do this just so that you can see what other people's experience were. We're still trying to build a semblance of community, even when we're all in different places. So if any of these words resonated with you, or it's just helpful to see what other people's experiences were, I hope that's something that you're getting out of this. And as always, I appreciate your willingness to share. All righty. So if you didn't get a chance to watch the Edpuzzle video on carbohydrate biochemistry, no worries. You can always go back and watch it before next time. I don't think you're going to be behind or in trouble for today, because I didn't technically assign any homework over the break. I think I posted that video yesterday with the hopes that you would have a chance to watch it for today's class. But if you didn't, feel free to watch it at your convenience. There's going to be a lot of chemistry in this part of the unit. So a lot of people are already responding. I have put up some slides in the Unit 6 slide deck that has some chemistry review that you might find helpful as we begin this part of the unit. But really just out of my own curiosity, but maybe you want to see what other people are thinking as well. Could you drag your icon to where your needs might be in terms of the chemistry review? I'll be very honest with you. I'm not going to be able to do that chemistry review with you in this setting. We do have some time, like from 2 to 3 p.m. This afternoon, if you wanted to meet up and ask some questions, but really just to get a sense of what your needs might be. So a lot of people have responded, kind of all over the place. Okay. If you chose, one of the higher scores, really kind of like from 5 up to 10, those links will take you directly to those slides that I've created that will give you a chance to review some of the chemistry that you might find helpful to better understand this part of the unit. So I would encourage you to take a look at those if those needs apply to you. Okay. Well, let's do a little bit of review. I'm going to give you control of the slides. Slides 130, 131, and then 132 have some review, some more practice questions based on the material that we covered last time. So I'll give you control of those. Okay. Which of course means if you needed to, you could go back and take a look at that functional group table if you don't remember any of the structures or any of the names of the functional groups. But just take a few minutes here as we get started. See if you can identify these functional groups by dragging your dot and then answering the next 2 questions that follow. And of course, I'm here for clarifying questions if that would be helpful. It's like most people have answered the first question. And about half of you folks have answered the second question, and we're just getting started on the third. Okay, take your time, no rush. You can go over any or all of these if that would be helpful. So just out of curiosity, if you can have your camera on, you can use your fingers. If not, something in the chat will be helpful. How much more time do you think you need to answer these 3 questions? Remember, a fist is 0. You can type 0 in the chat. Zeros, a couple of ones. Okay, take another minute. It's totally fine. Going to enjoy the seizure that Isaac's background is eliciting inside of my nervous system. All right. Would it be helpful to you to go over any of these or all of these just briefly?
Yeah.
Thumbs up if you've got your camera on. Thank you, Trevor. Mid yawn. It's a mindful yawn though, so it's fine. All righty. I'll take you out of student mode then, so we can look at things together. Bring my pen up here, and we can take a look. Do a different color. There we go. All right. So really functional group review on the first slide, just doing some identification. Let's see how you did. Oops. Come on, buddy. There we go. All right, so yeah, our blues, those are our amino functional groups. So those are the NH2 functional groups in this case. Looks good there. The carboxyl groups are indeed the C double bond to the oxygen, OH. Be careful, the OH there is not a hydroxyl. It's not attached to a carbon all by itself. It's attached to that carbon, which is double bound to the oxygen. So that makes this whole thing a carboxyl group. So nice identification there. And the last one was a functional group that I highlighted in the Edpuzzle video from last week, but we didn't do any practice with last week, called the sulfhydryl group, kind of a gimme, sulfhydryl, sulfur, S. So it looks like most people were able to identify the SH functional group at the bottom of that molecule. If you're wondering, this is an amino acid. It's a molecular building block. It's a monomer of a protein, the protein being a polymer, of course. And this SH is one of the parts of the amino acid cysteine. All right, looks good there. Well done. Moving on to the next one. Multiple choice. We are forming a bond. You're told that it's a peptide bond down here. So 2 monomers coming together to make a very short polymer, a 2 monomer long polymer. And ideally, this is showing a formation of a covalent bond. And I can see the answers are changing in real time. It's totally fine. These are not ionic bonds. They are covalent bonds. This is not a hydrogen bond. This is a bond between the carbon and the nitrogen. That is a peptide bond, but we'll be looking at that next week. But really what's most important, the gimme to this question, is the formation of water, right? In condensation or dehydration reactions, 2 monomers come together. Their functional groups react in a way where a new covalent bond forms between those monomers, and water is always a product. So nice work there. And then last but not least, again, kind of a gimme, same reaction, we're just naming the process. So hopefully everybody dragged their dot to the dehydration or condensation reaction. The hydrolysis reaction is the next reaction that we're going to look at here in just a minute. That's the catabolic process. That's the process that's used to take molecules apart and actually uses water to break a bond rather than producing water as a bond is formed when 2 monomers come together to make a larger biological molecule. Okay. So what questions does that bring up for you? Or are you ready for the next step? Thank you, Trevor. Appreciate it. All right. Dots are still moving. I like that. All right. So I hope that practice was helpful since we really haven't been together for a week. So indeed, if dehydration / condensation reactions put monomers together anabolically, right, so as to make larger biological molecules, then there should be a process that we can use to take those biological molecules apart through catabolism. And there is indeed a reaction, of course, and that reaction is called a hydrolysis reaction. So you've got a larger molecule. All those monomers that make up that larger molecule are linked together by covalent bonds. How are we going to take it apart? Well, we're going to use water, okay? And that water will come in. It's responsible for breaking that covalent bond between the monomers. In this case, we're just going to break this one bond. I can highlight it here. Okay. And what's going to happen is not only is water breaking the bond, but we're going to consume water in the reaction. So much so that you can see the water now becomes part of those now two separated molecules, right? This is catabolism. We're breaking molecules apart. We're accomplishing it through hydrolysis. Water is the reactant in this case. It's breaking the bond, but it's also being consumed in the reaction in a way where the water becomes parts of the molecules that have been created due to that break of the bond. Okay, so we now have our two processes, right? We can put molecules together through dehydration synthesis. We can take molecules apart through hydrolysis. So let's practice a bit. Okay. So enter a number type of problem. So if you had a polymer that was 150 monomers long, how many water molecules would you need to break that polymer up into its monomers? So completely break it down, to completely catabolize it. About half of you have a response. Remember, water is going to come in and break that bond to separate the molecules from each other. So if I had two molecules that were attached together, I would need one molecule of water to break that bond to form two monomers. If I had three monomers linked together, how many molecules of water would I need to break those bonds? If I had four, how many molecules of water would I need to break those bonds? All right, so most people have a response. Let's see what you came up with. 149. Looks like there were some answers over here in the 75 range. Okay, that's not going to be what we're looking for here. In reality, there's a couple simple ways to think about it. I guess you could draw out the polymer with 150 monomers all linked together and just count the number of bonds that have to be broken, but that seems awfully laborious. A really simple rule to help you remember something like this is if you know how many monomers are linked together in the polymer, in the larger molecule, then n minus 1, where n is the number of monomers, would equal the number of bonds that are linking those monomers together, and thus would give you the number of water molecules that have to be consumed, that have to be used to break those bonds. And there's some caveats. We're assuming the molecule is linear and not circular, but for the most part in biology, that's going to work. All right, let's try another one. Trying to connect some from the video, if you had a chance to watch it, on carbohydrate biochemistry. to get your response in. It's like 19 people have so far. That's awesome. So again, we're breaking molecules apart. This is hydrolysis is an example of catabolism. All right, let's see how you did. Yeah, absolutely, spot on. We're adding a water molecule, we're breaking a bond, and in this case, we're going to form two sugar monomers. And what those monomers are aren't relevant to us right now. But if you had a chance to watch the video, you know that there are lots of different possibilities. Glucose is the most common sugar monomer, but there's also fructose and galactose and other molecules that come together to make disaccharides like sucrose, good old table sugar, or even more complex carbohydrates like starch. So just keep in mind that hydrolysis breaks those molecules down. Beautiful. I think I've got one more for you. Much more coming up on enzymes next week and the following week, but really you don't have to know much about enzymes to answer this question. All right, let's see how you did. So we have a little bit of a mixed bag. But there's a leading vote getter that is quickly changing in real time. So amylase, as described in the question, is responsible for breaking down the carbohydrate. And the synthase molecule, not surprisingly, is going to be responsible for putting the molecules together. So indeed, B would be the correct answer because amylase is aiding in the addition of a water molecule to break bonds, whereas glycogen synthase is doing the opposite, right? It's going to be generating water as new bonds form. All right, well done. Again, I hope this practice helps. I put a question in the chat a few minutes ago about, do you get other opportunities to practice like this in other classes to get feedback? Because really this is what I want class to be. I don't want it just to be me lecturing for an hour or 45 minutes with whatever time that we do have. But again, inviting you to ask any questions that you have about any of this before we move forward. We will skip that one for now. Seems like a good time to take a short break. I've got 9:30 on my clock, so why don't we come back together at 9:33 and we'll delve into some real-life examples of carbohydrate biochemistry. I'll see you in a few minutes. Alrighty, we can start making our way back together. Alrighty. So everything is moving around here. All right, so welcome back. I hope you give yourself a chance just to stand up and stretch, get a drink of water, use the restroom. And as always, I hope you're getting breaks in other classes. I'm trying. If you did have a chance to watch the Edpuzzle video for today, you know that it was all about carbohydrate biochemistry. And really, carbohydrates are the first of 4 different types of biological molecules that we're going to encounter in this part of the unit. So there are fats. We'll talk more about fats next time. And then there are proteins. We'll spend really an entire week, or as much of a week as we can, looking at protein biochemistry. And then there are nucleic acids, that's DNA and RNA. RNA is a pretty hot topic now because both the Pfizer and Moderna coronavirus vaccines are based on RNA technology. So nucleic acids will actually come back to later in the year when we look at genetics and the transmission of hereditary material. But suffice it to say, there are 4 different types of biological molecules. We're going to start with carbohydrates. The connection, well, if we're going to put carbohydrates together, we're going to use dehydration/condensation reactions. If we're going to take apart carbohydrates, if we're going to break them down, we're going to use hydrolysis reactions. And now we have specific ways, right, to begin to think about those anabolic, endergonic processes that are responsible for putting molecules together. That's dehydration synthesis. But we also can think about how catabolism is accomplished within a biological system that's going to be accomplished through hydrolysis reactions. Alrighty. So I have kind of 3 different real life examples of why carbohydrate biochemistry matters. And I can give you a choice. One of them you were introduced to in the video if you watched it. You know, maybe you've heard of this molecule called Splenda or sucralose before, and we can look at specifically why sucralose, Splenda, is a calorie-free sweetener, if that is of interest to you. But I also have some other examples of why carbohydrate chemistry matters. One example is, why can cows eat grass, but you can't? And I guess you could eat grass, but you won't benefit, you won't get any of the benefits and nutritional benefits if you were to choose to eat grass. So where's your thinking lie? Simpler carbohydrates, kind of delving more into why things like sucralose doesn't have calories, or we're looking at some of the more complex carbohydrates be useful to you today? Just as real life examples, right? Not that you have to memorize them or you ever have to spit them back out on an assessment, but where do your interests lie? If you could put something in the chat or take yourself off mute just to give me some direction, that would be super helpful. Again, lots of examples. Do you prefer a simpler example of a carbohydrate or a more complex example of a carbohydrate? Thank you, Ryan. Thank you, Kellen. So far, you're the only 2 voters. Just so you know, I'm going to invalidate both of those votes. And then I'm going to do a recount. And I'm going to find that more votes were in favor of the leading candidate.
More complex.
Thanks, Trent. All right. So I'm seeing more votes for simple. I think we'll have time to do a simple and a complex. So I want to honor the request for people who had a request. Thank you, Ryan. I agree. So let's start with this example of Splenda. All right. So if you had a chance to watch the video, you were introduced to 3 of the more common disaccharides that are found within biological systems, within living systems. And let's be honest, these are also the molecules that we eat. By far, especially in a Western diet and an American diet, we preferentially are eating a lot of carbohydrates. And disaccharides are often the principal components of the carbohydrates that we eat. So of course, you got to memorize all the names of these carbohydrates, their molecular formulas, their structures, and where they're found, what they're composed of. No, of course you don't. That's ridiculous. That's not what this is about. These are just real life examples. So if we're going to talk about sucralose or Splenda, ultimately what we're talking about is a chemical derivative of the disaccharide sucrose. So sucrose as a disaccharide, a couple of quick things you might have noticed, but I think I asked in the Edpuzzle video as well. You might expect that if I were to put 2 monosaccharides together, 2 monomers together, C6H12O6 together, that I would have a molecule that was what, C12H24O12. But of course, because water is a product of that reaction, it is a dehydration reaction after all, I'm going to lose 2 of those hydrogens and 1 of those oxygens to form a molecule of H2O. Okay. The monosaccharides that are coming together to make sucrose are indeed glucose and fructose, but sucrose is just table sugar. It's white sugar. Heck, it's holiday cookie baking time, right? It's what's going into all of our baked goods this time of the year. It's by far one of the most common sweeteners found in foods today. But sucrose ain't sucralose. So the structure of sucralose, this is the same slide that was in the video, if you had a chance to watch it, is it has a very different structure than sucrose does. So sucrose is up here on the top. Maybe you're noticing some of the different hydroxyl groups that are part of its structure. There's quite a few of them. I'm not going to circle them all. If you look down at the structure of Splenda or sucralose, you're going to notice, and I think I highlighted this in the video, that some of those hydroxyl groups, 3 of them in fact, have been replaced by chlorine atoms. So just to kind of stimulate our thinking a little bit, and you may know the answer, you may not know the answer, take a moment to predict why you think sucrose, so not sucralose, but sucrose can be hydrolyzed. It can be broken down. It can be digested, if you will. But molecules like sucralose, like Splenda, can't. And then try to justify your prediction. It's almost like we're brainstorming. You may not know the correct answer right now. What is your thinking? We're eating these molecules, sucrose and sucralose. Sucrose can be hydrolyzed, it can be catabolized. But sucralose cannot. Why? Why might that be the case? Just take another minute to finish your thinking, to finish your writing. Oh, something popping up in the chat. Interesting, right? All right. It does not. Splenda does not raise blood sugar levels. It's a great sweetener for somebody who is pre-diabetic or even diabetic. All right, let's see what you came up with. Again, you may not know the answer. All right. Yeah, there's something to do with those chlorine atoms. It's not toxic, although some people do report that when they eat pure Splenda, so I'm thinking of those little yellow packets that, when we used to eat in restaurants and put things into our coffee or tea, they'll detect a slight chlorine aftertaste with when they put Splenda in their mouth, but it's not toxic. Really, the only difference between sucrose and sucralose is the presence of those chlorine atoms, where the hydroxyl groups would be in sucrose. Yeah, something to do maybe with the water molecule, can't be recognized. Yeah. Some would argue that sucrose is actually more complex than Splenda, but in reality, the only difference again is the presence of chlorine atoms in sucralose. Yeah, hydrolysis cannot occur with the chlorine. It must have only OH, hydroxyl. Yeah. Yeah, I think we're on to something. Well done. Thank you for sharing. Let me bring this back here. And a couple of things. All right. So it turns out, no, it's not going to work. There it is. Oh, come back. So it turns out that it absolutely has something to do with the structural differences between these two molecules, and specifically how another molecule inside of our bodies is interacting with those two different types of molecules. I'm talking about sucrose and sucralose, of course, because we can put either of those molecules into our body, right? We can ingest them, we can swallow them, they're going to move into our stomach. It's actually not where the digestion is going to take place in this case. Believe it or not, very little digestion actually takes place inside of our stomachs. Most of the digestive process takes place in our small intestine. It's where a lot of catabolism, a lot of hydrolysis is taking place. And there are molecules called enzymes. You may have heard of them before. If not, I don't think it's critically important to understand the excruciating details of enzyme structure and function to appreciate what's going on here. But what I have for you is a short video clip that's going to highlight how sucrose is hydrolyzed by an enzyme called sucrase inside of our bodies. Okay, so as you watch this, notice how sucrose is interacting with the enzyme sucrase, and then maybe think a little bit about how sucralose is interacting with the enzyme sucrase. I hope this will work on your end.
Sucrase resides on the surface of the microvilli on the intestinal epithelial mucosal cell surfaces. This animation presents a graphical representation of the way that sucrase catalyzes the hydrolysis of the common disaccharide sucrose, which we know as table sugar, into its component monosaccharides, glucose or blood sugar, and fructose or fruit sugar.
So hopefully you're seeing two things, right? The purple, that's the enzyme sucrase, and then sucrose is coming in, and it's physically interacting with the enzyme. It's binding to the enzyme. And as it does so, that water molecule came in, just like we would expect it to. This is a hydrolysis reaction after all, and breaking the sucrose into the two monomers. And again, you can memorize it if you want to, but the two monomers are glucose and fructose. They come together to make a molecule of sucrose. In this case, we're just taking it apart.
Hydrolysis is accomplished because when the sucrose molecule binds to the active site of the enzyme's configuration is changed so that the oxygen bridge between the two monosaccharides is exposed to water molecules in the solvent. This exposure permits a water molecule to actually break the bond, the oxygen bridge, and attach the components of water, an OH to one of the monosaccharides and an H to the oxygen, which is still attached to the second monosaccharide. This effectively cleaves the bond between the two monosaccharides and converts the disaccharide into two separate sugars. Once this is accomplished, the enzyme's configuration is changed back to the original shape. The two monosaccharides float away, and the site becomes available for another sucrose molecule to bind, change the enzyme's configuration, and be hydrolyzed. This action can be repeated many times until the enzyme becomes denatured, is inhibited, or just wears out.
Right. So given what you've just seen, if we go back to this question, do you want to revise your answer? Do you want to add something to your answer? You saw how sucrose is being hydrolyzed. Why might sucralose not be capable of undergoing hydrolysis? Just take one more minute. Think about if you want to revise your answer, change your answer based on what you've just seen. Yeah. And you can put it in the chat too if that's easier. So what's happening here is if there's an enzyme that's responsible for breaking down sucrose, right, for hydrolyzing sucrose into its two monomers, we can see that process happening as part of the animation, right? In the case of sucralose or Splenda, that isn't happening. And the reason why it's not happening is because if that enzyme sucrase tries to bond to a molecule of Splenda to sucralose, the two molecules don't bind to one another. They don't interact. And the reason why they don't interact is due to the presence of those chlorine atoms that have taken the place of the hydroxyl groups that are parts of the normal sucralose molecule. So if sucralose can't bind to the enzyme that hydrolyzes sucrose normally, well then it's not hydrolyzed. And what's left behind is a really big molecule. A big molecule that cannot be absorbed by the small intestine. It can't enter the bloodstream. So Ryan asked a question, I think he made it a question to everyone. Yeah, he did. Does Splenda raise blood sugar levels? Well, no, because it never gets into the blood. And it never really goes anywhere other than into the toilet. Eventually, you're just going to eliminate this, not even through your urine, because to make it up into your urine, it would still have to be absorbed into your bloodstream. It's just going to be eliminated through your poop. And here's what's nuts about this. About a decade ago now, some biologists wondered, well, wait a minute, is there Splenda in the real world? And when I say that, I don't mean are there living things that are making sucralose, right, that are making Splenda. Splenda is an industrial sweetener. It's a human-created sweetener, right? It's not found in nature, except it's found in nature because as we've eliminated this waste, some of that waste is finding its way into natural ecosystems in detectable levels. We put it there, maybe not on purpose, but we put it there by making something that cannot be broken down naturally, is eliminated, and finds its way into natural systems where it really doesn't belong. So aspartame is a different sweetener, a different artificial sweetener. It's actually derived from an amino acid, so the molecular building block of a protein that we'll be looking at later, aspartame aspartic acid. Yeah, when you start asking things like, could through evolution something eventually break it down? And that brings us really all the way back to the beginning of the year, right? In order for something like that to happen, right? There'd have to be a natural variant in some population, probably bacterial, but not necessarily bacterial, that has the ability, right, to recognize this molecule, but also has an enzyme that recognizes the structure of that molecule in a way that allows it to undergo hydrolysis. And we can't evolve the solution to that problem. We have to allow for natural selection to select for those individuals that possess that variation from birth. I'm talking about bacteria, I use birth in quotation marks, in order for that molecule to be hydrolyzed naturally. So the short answer is probably not, but it's not impossible. Okay. All right. Well, I want to honor the time that we have left because we are almost out of time. And just give you a moment to reflect here before you go. So molecular shape, molecular composition, it's kind of sort of important. These subcomponents of these biological molecules matter, hydroxyl versus chlorine. So how has your perspective on molecular shape, on molecular composition changed just based on what you experienced today? Add a sentence or two, and as soon as you have, you are welcome to sign off. Thanks for being here as always. Let's see.
Thank you.
You're welcome.
Thank you, Mr.
Take care, everybody.
Thank you. I love that. Parents won't let me have it. That's great.
Thank you. Bye.
Take care of it. Morning, Sarah. Whew. Crazy. Oh, you know, I don't really have an answer to that question. It's a simple question, right? I think I'm going to choose grateful. I'm grateful. How's life on your end? Yeah, I hear you. I feel like I have these windows of time where if I can just stay focused, I can get things done. But I know that if I don't get things done during that time, I won't have other time to get things done. So it promotes a sense of anxiety in me as well. Like, okay, come on, you gotta focus, you gotta focus. And it's like, but I can't focus, but you got it, but you got it, but you got it, but I can't. And I was doing my meditative practice this morning with a teacher who was, he's been working with professional athletes for much of his career. And he shared a sentiment that I thought speaks to this idea of anxiety. He said, that's exactly when you want to do the practice. It's when you want to step away from whatever it is that you're doing and ground yourself and reset, rather than trying to force yourself to focus, because that probably just doesn't work. Found that to be meaningful, at least for me. Here comes the folks. Good morning, everyone. Abby and Jesse, Lauren and Max.
Hi.
Do you still love me?
Always.
Always. Well, you know, we aim to be Bergeron Incorporated. Nona, good morning, Sasha. Ingrid, Alex, and Caitlin. Dawn, good morning. All right. Got just about everybody. Couple of quick things before we get started this morning. You may have got a text this morning from me through Remind about summative assessment number 5, still a take home. I think I'm going to just keep doing that for as long as forever. Seriously, I think it makes the most sense. I have the due date set for Monday, but please let me know if that timeline is not going to work for you. I'd much rather know ahead of time that I need to move it rather than force you into something that simply doesn't work for you. So please let me know if that doesn't work. And you can start that now if you want to, but I wanted to just say that I think there's one question, it's a multiple choice question that we're probably not gonna get to until Thursday. So just keep that in mind. By the weekend, it'll be fully ready to go. But if you hit that question, say tonight, if you were to start tonight, don't be alarmed, we're getting there. But you could still begin, leave the tab open, and come back to that assessment later on. Still pretty short. I think I've got 8 multiple choice questions and 3 free response questions. So it shouldn't take you too long regardless. Second item, maybe because I'm a crazy person, but maybe not. The next time that we get together, which is supposed to be an in-person experience for at least some of you, I thought we could try a really simple lab experiment. If you're at home, no worries. I have a virtual equivalent for you to take a look at. But I thought we could try it. Of course, socially distanced, all that good stuff is going to be in full effect. But that is part of the agenda for next time. And yeah, it's simple. Really, I want to try it just to make sure that it's something that we can do before we try something more complex later on in the semester and really for the rest of the year. So that is going to be part of the plan for Thursday. But as always, opening the floor to questions.
Wait, I have a question. So do we do the virtual lab that you posted, or is that what we're going to do in class?
The lab number 8 is the lab that I'm hoping we'll have, if you're going to be here in person, right, that we'll have time to do on Thursday for this class. If you're not going to be in school, you could do that now, right? If you wanted to, as the replacement. I'll leave that up to you to decide what works best in terms of your schedule. But yes, lab number 8 is the lab that we're going to try to do in person. But what you see on Classroom is the virtual replacement, the virtual equivalent for those folks who are not coming to school.
Okay, thanks.
Other questions? Okay. All right. Well, then our plan, I've got a quick check-in for us this morning because it has been a week since we have been together. And I've got some review questions on where we left off last time, thinking about the biological functional groups and putting biological molecules together anabolically through dehydration/condensation synthesis reactions. And then I've got the other half of putting molecules together. That is, of course, taking molecules apart catabolically. So I thought we could look at the reverse of condensation. That's a hydrolysis reaction. And I've got some practice questions for us to look at there as well. And then with whatever time that we have left, my hope is you had a chance to watch the Edpuzzle on carbohydrate biochemistry for today. And I've got some other real life examples of why carbohydrate biochemistry matters. Not because you need to memorize them, not because they're going to be on the test, but hopefully because they'll just kind of stimulate your interest a little bit about all these molecules that really, as Americans, as Westerners, make up a huge part of what we put into our mouths every single day. So that is our plan for the day. Since it has been a week since we've been together, and over that week there was a holiday, whether you celebrated it or not, we still had a break. My hope is that you could take a moment this morning, reflect over these past few days, and in a word, you can write it, you can draw it. What word best represents your experience over your time away from school? And if you need more than looks like most people have had a chance to write something, so I thought we could just take a look at what people's experiences were. Yours are going to be at the bottom here. Fifth period is going to be at the top, so we'll kind of go in reverse. Food, yeah. Stressful. Tiring. Calm. I'm going to assume that's a turkey. But yes, necessary. Relaxing. Change. It was another example of change, wasn't it? Enjoyable. Relaxing. Relaxing again. Carnage, hopefully for the turkey and the sweet potatoes and the loaves of bread that were turned into stuffing and not, family members. Relaxing, relieving. That's the first time I've seen that. Anxiety, overwhelming, stress, man. Productive, short, on edge. Okay. Well, looks like there was kind of a mixed bag of experiences, and that's okay. My hope is that whatever you wrote you saw something that you were able to connect to, whether it was the same or different. You know, I often come back to the word weird when people ask me how things are going at school or how things are going in life or how Thanksgiving break was. It was just weird, right? You couldn't see the people that you normally see, but you could see some people. And you hear about the people who traveled on airplanes and what that must have been like for them. Weird, I would assume, but maybe not. So regardless of what word you chose, thank you for sharing. Thank you for reflecting. And again, we're trying to build a sense of community, right? Of shared experiences, even though we're not in the same place. So I hope that you find this at least a little bit useful this morning. Alrighty. You've probably gathered at this point that this part of Unit 6 is all about biochemistry. So there's the root, chemistry, an experience that you all had last year or maybe 2 years ago if you're a 12th grader. Really, I'm just curious. I can't necessarily give this to you given the constraints of the time that we have together, but where are you right now in terms of your knowledge of general chemistry? Could you quickly drag your dot, or the icon, sorry, in this case, to your needs in terms of the general chemistry review? This is a very busy slide. So kind of all over the place, but maybe more concentrated in the middle. So to honor the middle and maybe even the higher numbers on the right-hand side, I did want to say briefly that the two prompts that are hyperlinks, they will take you to some slides that I made that I think, I hope, will serve as a little bit of chemistry review. Really like the bare bones, what I hope you learned in chemistry that applies to biology right now. So if you ever want to go back and click on those links, it'll take you right to the slides that I've created, and you can use those as a way to review some of the chemistry that you learned a year or two ago. I'm really sorry that we don't have time in class to go over that, but I will say that at least for today, and certainly by appointment, if that would be helpful, we have that hour from 2 to 3 p.m. this afternoon where you're welcome to stop by and ask chemistry questions. And hopefully, we'll build some time into the days moving forward where you can ask those questions as well. Yeah. I'm looking at the chat and I'm seeing what you're writing. So we're going to take it slow. We're going to take it slow. And my hope is that you'll ask lots of questions if you have them. And you'll use those slides that I've created just as a way to review. And the lectures, if you will, the new content, I'm sure you realize by now, are all coming in the form of Edpuzzle videos, right? Where you can always go back and watch them again if you need to. So we'll do this together as slowly as we need to. But thank you for offering your responses. Well then, in the spirit of review, thinking back to last week, we did a little bit of work with functional groups and how they relate to biological molecules. And we had some time to look at the one type of reaction, I think we got that far in this class, that puts biological molecules together as new bonds form between the functional groups that are parts of those molecules. So I just got 3 quick review questions for you to look at. They're on slides 130, 131, and 132. So you are now in student mode. Take just these next few minutes. See if you can identify the functional groups, and then answer those next 2 questions that connect to putting biological molecules together anabolically through synthetic processes. And of course, I'm here for clarifying questions if needed. Looks like the first question has been answered by most folks. So keep working on the next two. No rush. Remember, since you're in student mode, you can always go back and look at the functional group chart, the functional group table, if that would be helpful. All right, we're getting there. All right, it looks like just about everybody has submitted a response. Does anyone need some more time? You can show me a finger minute or pop in the chat. Remember, a fist is 0 if you have your camera on. You type 0 if you don't need any more time. Thank you, folks. Helps me to know, A, that you're there, B, that I'm not going too fast. All right. Would it be helpful to go over some or all of these review questions? Give me a thumbs up, either in real life or electronically. Okay. That's kind of what I figured. All right. So let me take you out of student mode then, and we'll take a look at what you have come up with. So first one, again, we're just trying to identify some of the functional groups that are part of larger biological molecules, because these are the parts of molecules that are going to react in these reactions, right? In these dehydration or condensation reactions. And spoiler alert, they're also going to be the parts of the molecules that react when molecules are taken apart catabolically. So we've got 3 functional groups here. Let's see how you did. Blue dot is your amino group. Beautiful. Orange dot, the carboxyl group. Looks very nice. And the green dot actually was one that we didn't look at last time, but was part of the Edpuzzle video, the sulfhydryl group. In green is down at the bottom of the molecule. So really well done. And just, I'm going to keep saying it like a Spotify playlist on repeat. You don't need to memorize these. Right? Print out, screenshot, keep a link to that slide on your desktop. You can always go back and look at it whenever you need to. Alrighty. Next one, which of the following best describes the formation of the bond shown in figure 1? You're told down below that it's a peptide bond. That doesn't really help you to answer the question. But ideally, what you're beginning to see, at least a little bit here, if I pull up my pen, is we've got these two molecules, these two monomers at the top of the image. And it looks like we're going to be linking these molecules together end to end, right? And as we do so, we're going to form a new chemical bond between them. So here's that, I'll change colors here, so it's a little bit easier to see. Here's that C double bond O, that's part of the molecule on the left. And here's the NH, sorry, that's part of the molecule on the right, okay? So we're linking those molecules together. Let's see what you came up with. Absolutely, right? You want to choose the answer that describes the dehydration or the condensation reaction where water is a product of the reaction. Okay, very nice. Clear that off. One more. This one was, I'm hoping, the easiest of the three. What term is used to describe the process in the image? Well, again, this is dehydration, condensation, synthesis. We're taking two monomers and we're linking them together with a new covalent bond, and we're building a larger molecule. In this case, a very short polymer. It's only composed of two monomers, but that is an example of dehydration synthesis. And looks like most people correctly identified the process as dehydration, so way to go. All right, is this bringing up any new questions for you or anything you're still uncertain about before we go on? Well then, so all of those review questions dealt with putting molecules together, right? So that's our anabolic synthetic process when we think about putting molecules together. And of course, those molecules, because they're being synthesized anabolically, would have more chemical potential energy associated with them than do either of their reactants. But there has to be a reverse process, right? If metabolism has an anabolic and a catabolic side, there should be a biological reaction that allows for molecules to be catabolized or to be broken down. And in fact, there is. These are the hydrolysis reactions. So if condensation reactions, if dehydration reactions put molecules together, then hydrolysis reactions take molecules apart. And still a pretty simple process. And in reality is just the opposite of dehydration. So in this example, we've got a short polymer. Nothing fancy, we don't know what it is. Looks like it's composed of 4 monomers though. Monomer 1, monomer 2, monomer 3, monomer 4. And I changed my color real quick. We've got 3 bonds that connect those 4 monomers together. And in our image, we're gonna break, we're gonna catabolize, we're gonna hydrolyze this short polymer into, well, a slightly shorter polymer and 1 new monomer. So monomer, 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 and then here's now this free monomer that's been released from the larger polymer as a result of the hydrolysis reaction. Well, how is it happening? Simply put, water is attacking the chemical bond, change colors one more time, between 2 of the monomers. It's causing that bond to break, which is releasing the monomer that we see over here. But the water also is being consumed in the reaction. How do we know? Well, as water attacked that bond, it too was catabolized, it too was broken down. And here are its constituents, right? H2O. Okay, so unlike dehydration synthesis, hydrolysis is taking molecules apart. In dehydration synthesis, we generated a molecule of water. It was a product of the reaction. In hydrolysis, water is a reactant in the process and allows for molecules to be broken down rather than synthesized. Okay. So, questions, wonderings before we do some practice? I have 3 practice questions. Erase this so you can see it. If you had a polymer that was 150 monomers long, how many molecules of water would need to be consumed to break that polymer all the way down into its monomers? Go ahead and type your number when you think you have your answer. All right, let's see what you came up with. Not sure why somebody decided to put such a large number in there. But indeed, it looks like most of the answers, it's out of proportion, are hovering around the 149, which is the correct answer, right? We need a molecule of water to break every bond that's linking all those monomers together. If there's 150 monomers that make up this larger polymer, that means there's 149 bonds, covalent bonds, linking all of those monomers together. So we'd have to invest 149 molecules of water to break all those bonds and release all of those monomers into solution. Yeah, that might be an important point too. Like all of these molecules, all of these reactions, they're taking place in water, right? So when we say things like we're investing a molecule of water or we're producing a molecule of water, there is plenty of water to go around, right? Water is never going to be a limiting reagent in any of these processes. All right, let's just try one more. This is actually an AP free response question. Actually, I might have 2 more. Take that back. This connects to the Edpuzzle video a little bit. Again, it's practice. If you have clarifying questions, you can pop something up in the chat. All right, let's take a look at what you came up with. Yeah, right. If we had a carbohydrate, we don't know what it is. It could be any of the ones that you were introduced to in the Edpuzzle video, which of course you have to memorize, right? All the molecular formulas, all the structures, all their names, or else you're going to fail in life. It's being ridiculous. Yeah, we're talking about breaking down molecules into their monomers, carbohydrates or sugars. So absolutely, we're adding a molecule, it's breaking the bond, we're releasing the monomers. Very nice. Now I think I have one more for you. Again, another AP practice question. you know nothing about these enzymes, I've probably never heard of them before, you don't have to. Got kicked off. Let's see. Oh, there she is. One converts polymers into monomers, the other converts monomers into polymers. Right, taking a look one more time at your responses. Looks like we have a clear leader in terms of votes, which means we probably should do a recount just to see if more votes for the leader magically appear. Yeah, right. Amylase is converting polymers into monomers, right? So we're adding a water molecule to break bonds, whereas the other enzyme that's putting monomers in together to form polymers is doing the opposite, right? It's going to be generating a molecule of water through dehydration rather than using a molecule of water as part of a hydrolysis reaction. All right. Well done. So I thought this would be a good time to take a short break. I've got 10:33 on my clock. Why don't we come back together at 10:36, a nice 3-minute break, and we'll look at carbohydrate biochemistry just a little bit more. if we could start making our way back together. All right. So as we get a little bit more specific in terms of biochemistry, there are 4 different types of biological molecules that we're going to be studying these next couple of weeks. Carbohydrates are one of them. And again, I hope you had a chance to look at the Edpuzzle video on carbohydrate biochemistry. Fats, lipids are the second category, proteins are the third, and nucleic acids are the fourth. Of course, nucleic acids are really all over the news media right about now because both the Pfizer and the Moderna coronavirus vaccines are based on RNA technology. So these molecules are showing up in our real lives. And although we're starting with carbohydrates, my hope is for these next 15 minutes of class or so, I can provide you with some real life examples that show why these molecules matter to us. And hopefully, you know, maybe even prompt your interest a little bit. So I have several different examples that we can look at, ranging from the simpler of the carbohydrates. So I'm thinking of the monosaccharides and the disaccharides. But I also have an example of why the structure of more complex carbohydrates matters. These are the polysaccharides, of course. So really just to kind of probe your interest here, you can say it out loud, you can pop it in the chat. Where do your interests lie this morning? Would you like to look at a real life example of why simple carbohydrates matter? Or would you prefer to look at a real life example of why one of the larger, more complex carbohydrates matters? You could just put something up in the chat or say something out loud. That would be really, really helpful to me. Looking for a leading vote getter, but some people said either. And then it's a 1-to-1 versus simple to more complex. So maybe I'll choose. Maybe you don't have a preference. That's okay too. Well, why don't we look at one of the ones that was actually in the Edpuzzle video, but unpack it just a little bit further. Alrighty. So if you had a chance to watch the video, you know that there are 3 general categories of carbohydrates. There's the monosaccharides, which are the simplest of sugars. There are disaccharides, which are a little bit more complex. They're just composed of 2 monosaccharides linked together. And there are the polysaccharides, which could literally constitute thousands of monosaccharides that have all been linked together. And you need to memorize, you know, names and molecular formulas and the structures and where they're found in order to be, you know, successful. No. The example that I highlighted in the video in terms of a simple carbohydrate actually was Splenda or sucralose, if you're going to use the chemical name. And sucrose is white sugar. It's table sugar. Heck, it's holiday time. Maybe you're already making cookies or cakes or some sort of baked good in your own homes. And invariably, you're going to be using sucrose. Even brown sugar is just white sugar with molasses added to it to make it brown. So we as Westerners, as you know, as Americans, are eating a lot of sugar, and when we eat sugar, we're eating mostly sucrose. But sucralose or Splenda is a very different molecule, and I highlighted some of the differences in the video. But really, as we get started with this real life example, if you look at these 2 molecules, they're very similar in terms of their structure. Sucrose has all these hydroxyl groups. not going to circle them all, associated with its structure. But Splenda, or sucralose, has had some of these hydroxyl groups replaced with, of all things, chlorine atoms. So what I'd like you to do before I show you the answer, because you may not know right now, that's okay, can you predict why molecules like sucrose can be hydrolyzed, can be catabolized, can be broken down by adding a molecule of water, breaking a covalent bond, and releasing those two monomers from each other? But sucralose can't. And then can you offer a justification for your prediction? So really just maybe 2 sentences. Why can one molecule be hydrolyzed, but the other can't? And why might that be the case? It's okay if you're not sure. What do you think? All right. Let's see what you came up with. Here, and your answers are going to be at the bottom. Definitely has something to do with the chlorine atoms. Careful, these are chlorine atoms, not molecules. Chlorine, really sucralose isn't any bigger than sucrose is. I guess you could argue that it's a little bit more massive because chlorine is going to have a larger atomic mass than oxygen and hydrogen put together. But in terms of its three-dimensional size, it's not that much bigger. Super stable molecules, those little white packages. Remember those days we used to go to restaurants and there'd be little sweetener packets out on the table that we could add to our coffee or tea or whatever our beverage. Super stable, right? Can sit out at room temperature, no degradation. Yeah. There's certainly something about the body not being able to break down the molecule. Yeah. Okay, I like your thinking. Let's take a look at what is actually happening here. And to better understand what's happening in the context of sucralose or Splenda, I thought I could show you a short animation of what happens in our bodies to sucrose. So this is good old white sugar, table sugar, it's brown sugar as well. I mean, well, I kid you not, brown sugar is white sugar with molasses added to it to make it brown. But what you're going to see is the The enzyme, the molecule in purple, is called sucrase. That's responsible for hydrolyzing sucrose after we eat it. And indeed, this enzyme sucrase, the molecule that's doing the hydrolyzing, is found within our small intestines. So as I press play, just kind of watch and observe what's happening to sucrose as sucrase, the enzyme, interacts with it.
Sucrase resides on the surface of the microvilli on the intestinal epithelial mucosal cell surfaces. This animation presents a graphical representation of the way that sucrase catalyzes the hydrolysis of the common disaccharide sucrose, which we know as table sugar, into its component monosaccharides, glucose or blood sugar, and fructose or fruit sugar. Hydrolysis is accomplished because when the sucrose molecule binds to the active site of the enzyme's configuration is changed so that the oxygen bridge between the two monosaccharides is exposed to water molecules in the solvent. This exposure permits a water molecule to actually break the bond, the oxygen bridge, and attach the components of water, an OH to one of the monosaccharides and an H to the oxygen, which is still attached to the second monosaccharide. This effectively cleaves the bond between the two monosaccharides and converts the disaccharide into two separate sugars.
So I'm going to pause it right there. What I'm hoping you're noticing, right, is first, there's a physical interaction between the enzyme in purple, sucrase, and the molecule of sucrose. And that sucrose is binding to sucrase. So there's a direct physical interaction between those two molecules. And as a consequence of that interaction, a molecule of water is able to break the bond. This is hydrolysis after all. And the water is consumed. It's a little hard to see, but not impossible. There's an OH being regenerated and an H being added to the fructose molecule. That's where the water is going. And then the enzyme releases those two monomers, right? They were once part of a larger disaccharide, sucrose, but after hydrolysis has taken place, those two molecules now are free from one another and they float away from the enzyme. And in fact, the enzyme can continue that process again and again and again and again and again until, I mean, you can imagine, all the sucrose has been converted into glucose and fructose. Sucralose, Splenda, with those chlorine atoms, and there's just 3 of them, right? 3 atoms have taken the place of 3 of those hydroxyl groups. Sucralose can't interact. It can't bind to the enzyme sucrase. The 2 molecules will not bind to each other. Those little tiny differences, those subcomponents of sucralose, of Splenda, the chlorine atoms that aren't part of sucrose's structure with its hydroxyl groups, prevents molecules of sucralose from binding to the enzyme. So we've put these molecules into our body, right? They've entered our mouth. We've swallowed them. They go into our stomach. In reality, not a lot of digestion takes place in our stomach. Most of the digestive process takes place in our small intestine. And this is where sucrase resides, which is great for breaking down, for catabolizing, for hydrolyzing sucrose. But Splenda, sucralose, not so much. It can't be hydrolyzed. It can't be catabolized. It can't be broken down because those chlorine atoms prevent it from physically interacting with the enzyme. So where does it go? Well, because it can't be broken down, it's never absorbed. It never even enters our bloodstream. So Splenda is this amazing molecule, right? We can put it on our tongues. It tastes sweet. It tastes like sugar, and I'll offer the caveat to that. Some people actually report a slight aversive aftertaste when they put Splenda in their mouth. Well, guess what, folks? That's due to the presence of those chlorine atoms. But regardless, it still tastes pretty sweet, but it goes down. It's never broken down. It's never absorbed into our bodies, which means it's eliminated from our bodies when we poop. And here's what's nuts. About 10 years ago now, a group of biologists wondered, can we find Splenda in nature? Because there isn't any living thing that's making sucralose, that's making Splenda, except for humans, and we make that in industrial laboratories for the purposes of sweetening things artificially. They found it. They found artificial sweetener. They found Splenda in natural ecosystems. We put it there. Now, the organisms that live there, they don't know what to do with it either. Now, don't get me wrong, there aren't piles of Splenda accumulating next to riverbanks, right, in these systems, but they were able to detect measurable amounts of this artificial sweetener, which raised all sorts of questions about, pollution that we never thought was possible, because we never really considered the possibility that something that tastes sweet but can't be broken down would ever end up anywhere other than the toilet. But it's there, and we found it. And the question becomes, how will organisms respond to it in the future? So I hope that provides a real life example of why the structure of these molecules matter, why the subcomponents of these molecules matter. It's going to be a recurring theme in all of the molecules that we study in this class. And heck, it's exactly how these vaccines are working as well, right? We're trying to train our immune system to recognize the subcomponents of a virus that is infecting the human population right now, so that our immune systems will recognize the part of that virus and mount an attack against it. It's real life. That's not carbohydrate, that's protein biochemistry, but still, the same idea applies. All right. Before you go, if you would, just take a moment to reflect. And I'll pull up the exit slip here. How has your understanding of molecular shape or molecular composition changed after doing what you did today, hearing what you heard today, seeing what you saw today? If you could just take a moment, write a sentence or two. Afterwards, you are welcome to sign off, and I will see you in a couple of days.
Thank you. Love you. Bye.
Take care.
Thank you.
Be well, everybody. See you in a couple of days.
Bye. Thank you.
So slow.